Okay, so this is the last one. This is question 6 for the 2023 FRQs. This one is looking like we're going to have to analyze this box and whisker plot. Okay, so this one says housekeeping genes encode proteins involved in universally important processes such as transcription, translation, and glycolysis. Because these genes appear to be expressed in all cells at constant levels, the expression of housekeeping genes is often used as a control when comparing how the expression of other genes vary under different conditions. Researchers studying the effect of pesticides on declining bee populations wanted to determine whether the expression of 4 housekeeping genes, GAPDH, RPL32, RPS5, and TBPAF, was in fact constant in bees across different variables. The researchers collected samples of mRNA for each of the 4 genes and compared how their expression varied across the developmental stage of the bee, the sex of the bee, and the cell type from which the sample was taken. The mRNA from the samples was reverse transcribed to produce DNA copies of each gene. PCR was then used to amplify the DNA, and the CQ values were determined. The CQ value is the number of PCR cycles needed to produce a specific number of DNA copies. A high CQ value for a sample indicates the gene was expressed at low levels. To analyze whether any of the examined variables affected expression of the housekeeping genes, researchers examined the range of CQ values for each gene in response to each variable. Genes with a wide range of CQ values were determined to be affected by the variable, while genes with a narrow range of CQ values were determined to be unaffected by the variable. Okay, and then this is our graph here. So this is probably a new type of graph, and this is including new types of measurements, which include CQ values, but they do tell you in the question everything you need to know. Okay, so what this question is saying is they're analyzing the expression of 4 genes, and these are the genes right here. Okay, they are trying to see if they differ between developmental stage, sex, and cell types. Okay, so they looked at the expression of all 4 genes per developmental stage, per sex, and per cell type. Okay, and they wanted to see how much it differs. The way they measured that is using CQ values, and this gives you the background on basically qPCR and how you determine expression. But really the most important thing for this is right here. High CQ value indicates the gene was expressed at low levels, okay? So that is going to allow you to determine whether these are highly expressed or have a low expression. And then genes with a wide range of CQ values were determined to be affected by the variable. Because if there is a wide range of CQ values, that means there's a wide range of expression, okay? Therefore, the variable is playing a role in the expression, right? If there is a really wide range of, GAPDH expression between developmental stage, sex, and cell types, that means that the sex affects GAPDH expression and cell type affects GAPDH expression. If there is a very small range, meaning that the expression stays the same between developmental stage, sex, and cell types, that means that it's not affecting. So that, sex does not affect the expression. So we're looking at if there's a wide range or a small range, and then we're also looking at the CQ values. So this is known as a box and whisker plot, and this isn't a very common type of graph that you're going to see, but you do need to know how to read these. So the way that these are done is this is not one measurement. This is, probably hundreds of measurements, and we're averaging them out. So this spread that you're seeing here, let me grab a different color. This top part of the box is representing the first quartile. The bottom of the box is representing the third quartile. And these error bars here are representing everything past that. And everything past those error bars are going to be considered outliers. The line that is in the middle of these boxes are the medians, okay? So it's showing you the distribution of the data. if I have data up here, so ranging, from what looks like 26 to 19, that's a very large range, right? So I have values, going from 26 to 19, however, the median is sitting around 20. But for this one, it's pretty much that I'm ranging between maybe 21 and 22. So this one has a very small range. That means all the values are pretty much centered and condensed around 21 and 22. So the smaller the box, the more constant the data, okay? If there is a wide range of data, that means that you are getting data that is, really high values, really low values, and then again, that line is going to be the median. So A is asking, based on figure 1, identify the gene that has the lowest median CQ values when Bs of different developmental stages were compared. Okay, so it's just asking you to identify the gene. So we're looking for the lowest median CQ value when Bs of different developmental stages were compared. So they're not asking you between all 3 of these variables, they're only asking you for developmental stage. So when I compare developmental stage, how does the expression change? And which one has the lowest median CQ value? So which one is the lowest? So in this case, we're looking only at developmental stage. The one that is the lowest is this one right here, the lowest median CQ value. Again, this one is the lowest regardless, but the median is right here. So this median is lower than this one, and then this one here. So that one, using our legend, this like kind of lighter gray colored in one is RPS5. So that one is going to have the lowest CQ value for developmental stage. So A is PS5. It's just asking you to identify. So when it says identify, you don't need to explain why. You just need to put it down. Okay, B says the CQ value is inversely proportional to the amount of mRNA from the gene in the starting sample. We already know this because they told us that in the question. It's inversely related, right? The higher the CQ value, the lower the amount of expression. Okay? Based on the data in figure 1, identify the gene that has the lowest level of gene expression regardless of variable. Okay, so this is asking for us to identify a gene again that has the lowest level of expression regardless of variable. So across all 3 categories. So again, lowest level of gene expression is not lowest CQ value. It's an inverse relationship. So the gene with the lowest gene expression should have the highest CQ value. So what we're actually doing is looking for the gene that has the highest CQ value consistently through all of the different variables that we're looking at, okay? Again, it's an inverse relationship, so lowest level of gene expression should have the highest CQ value. Let's look at which one has the highest CQ value for all 3 of our variables. In this case, it's this one, and this one, right? If I were to compare the medians, median, and then the medians here. That is the highest across all 3 different types of variables. This gene is TBPAF. So since this one is the highest across all of the different variables, so developmental stage, sex, and cell types, it is the lowest gene expression. They have the highest CQ value, therefore the lowest gene expression. So B is, again, it just says identify, so you just need to put down the name. It's TBP-AF. Okay, so that's it for question B. Okay, so part C says the scientists investigated the effect of pesticides on the expression of other genes in one cell type of a group of bees containing males and females of the same developmental stage. They hypothesized that TBPAF would serve as the best control gene for this experiment. Use the data to evaluate their hypothesis. Okay. So they are looking at the effect of pesticides in one cell type, so we're not using cell type as a variable, of a group of bees containing males and females. So we are comparing sex here, males versus females. They do have the same developmental stage. So that means that the only thing that's changing here is between males and females, okay? Developmental stage is the same, cell type is the same, you're only looking at sex. So they're saying to analyze the effect of pesticides per sex, that TBPAF is the best control. So the data that we're looking at in this case is just for sex. So we're only looking at this. They are asking you to justify why TBPAF, why this one, would be the best control. Okay? So you have to think about what makes a good control, right? We choose controls based on how consistent they are. Okay? So you use your control as a reference. You are comparing, this back to your control. And, you're comparing another thing back to your control. If your control is, giving you values that are at a super wide range, it's not a good control. So, like I said, the range of CQ values determines how spread out the data is, right? Because this is not one data point, this is probably hundreds of data points. So, a very small box, meaning a very small range of CQ values, is gonna mean that the gene is pretty constantly expressed. And that's what you want for a control. For a control, you want to find a gene that is pretty constantly expressed so it serves as a good reference point, right? In this case, it does not matter what the CQ value is, because either way, you're going to be comparing and you're going to be subtracting. What does matter is how spread out the data is. So in this case, they're saying TBPAF would be the best control, and I would agree with that because TBPAF has the narrowest range of CQ value, or basically the smallest box. Okay? It doesn't matter that it's the highest one, meaning it has the lowest expression. It has the most consistent expression. So this one over here, GAPDH, is fairly spread out, and these ones are pretty close as well. I would say TBPAF is definitely the smallest box and the most narrow range of CQ values. That means when they tested this, it gave you the most consistent expression levels. You do not want to use something where the expression levels are all over the place, right? These are really large boxes, okay? So you always want to use something that stays consistent if you're using it as a control, which is, again, the point of a control. So for this one, I would say that TBPAF would be a good control because it has the smallest range of CQ values. It is the most consistent. It does not matter that it is the highest value because, again, you're just using it as a comparison, and that comparison is going to stay the same throughout, okay? So it doesn't matter, like, let's say, I'm just trying to explain why, because I have a lot of students say, oh, because it's, the lowest expressed. That does not matter. You're looking for consistency. So let's say this one's, around 21, right? I am comparing everything to 21, okay? So no matter whether my CQ value is, 25 or 19, I'm comparing that to 21. However, if I have a really wide range and I'm getting, values between 20 and 23, and then I have 25 and 19 as my values that I collected, this is not very good to reference from, okay? Because am I comparing it to 20? Am I comparing it to 21, 22, 23? There's a wide range, so it's not a good control. If it stays relatively consistent, in this case, around 21, and again, there is going to be a little bit of variance just because of human error and system error. But again, what you want for your control is the most consistent one, okay? So for this one, C, I would say that TBPAF would be a good control, or the best control out of the four. So there's a couple different ways that you can say this. The way that I would say it is probably that it has the most constant expression. You could also say it has the smallest range of CQ values. It means the same thing, they're just different metrics. The best control, because it has most constant expression. Slash, so you didn't have to say both of these, they both mean the same thing, they accepted either. Most constant expression or narrow CQ range. Okay, that's C. Now our final one for all these FRQ questions for the 2019 one is explain how expression of a gene such as GAPDH can vary from one cell type to another within the same bee. So they are asking you why GAPDH can vary so much between cell types, okay? And again, within the same bee. We touched on this on a previous FRQ question within this exam, but again, it's the idea of all of your cells are genetically identical. So within the same bee, all of the cells are genetically identical. Why do some cells express GAPDH more than others? What's causing that? Okay, and there's a couple different things that you can say for this, but really it's about differential expression. Why is, GAPDH maybe more expressed in heart cells than it is in brain cells, despite the fact that they have the exact same DNA? Okay, it is going to be determined by what's going to regulate your expression. So you can probably say a couple different things for this, but I would say the easiest answer would be talking about transcription factors, right? So that transcription is going to determine your expression, right? So different cells have, you can either say different transcription factors or different concentrations / amounts of transcription factors. So let's just say different levels. Different cells have different levels of transcription factors. And again, transcription factors are going to help facilitate transcription, aka help facilitate gene expression. Transcription factors affecting expression. So that's why there's going to be different expression levels between different cell types, is because different cell types have different levels of transcription factors. That the changing or the different levels of transcription factors is what makes cells specialized. That's what makes your heart cell a heart cell and your brain cell a brain cell. The DNA is exactly the same. It's the expression of the genes that's different, and that's the expression of the genes that make it specialized to its job. So different cells have different levels of transcription factors affecting expression of genes. Okay. Alrighty, so that is it for all the questions for the 2023 FRQ. We finished off with this one right here. It's also out of 4, so you get 1 mark for each. So hopefully this was helpful, and I will see you on the next FRQ review session.